Hello and welcome to the Christians in Science annual conference. This is day 2, and if you missed day 1, that was yesterday, you can catch up on our website. Well, a special welcome to you today. If you're coming back, welcome again, and especially if you're not a member of Christians in Science and you're new to us. And we hope you and everyone will enjoy each of the talks this week and find the conference worthwhile. Now, we at Christians in Science are a network of mostly professional scientists, but it includes philosophers and theologians and engineers and teachers in schools and anyone interested in the science and religion interface. So you can find out more about us on our website, and I encourage you to look there afterwards. And there you'll find more about what we do and what we can offer in terms of resources. So our conference this year is online for the first time as a result of the pandemic. And of course, the theme is our response as Christians to the situation. After each talk, we'll be having a question and answer session with the speaker. And so we encourage you, as you listen and form questions, to send them to us using the comments box in the bottom of your screen if you're watching on YouTube, or the similar box if you're watching through Facebook. We'll also be having a panel session on Friday where we'll bring all of the speakers together to answer your questions, and you can send extra questions in between now and then, and we'll do our best to include them, and especially if we don't get your question answered on the day. So please do feel free to send those questions in to us. And you realize, of course, that this conference is now online and open to everybody, and so we haven't been charging a registration fee. And of course, you haven't had to pay a rail fare to get to London to meet together, which is unfortunate, of course, but it does save us that wee bit of money. And so I hope you won't mind if I suggest you might make a donation to help cover the costs of the conference and our ongoing costs at Christians in Science. And you can do that by text using the numbers on the screen below there. If you want to give by another means, you can find other ways of doing so on our website. So the other thing we'd like you to do, if you will, especially if you're new to Christians in Science, is to let us know your comments on the conference this week, whether you've just come for one day or you've listened to all of the sessions. We really value your comments that you could send to us, again, using the feedback form that you'll find on the YouTube channel description of the broadcasts. You also find it on social media like Facebook, and also, of course, on our website. So I do encourage you to let us have your thoughts on whether you find the conference useful or anything we might do to improve things. So today we come to our second talk in the series, and our speaker today is Dr. Miriam Schilling. And Miriam received her PhD from the University of Freiburg, the Institute of Virology in Germany, in 2016. And she's currently working in Oxford doing postdoctoral research in the interplay of viruses and the innate immune systems. In her spare time, she is also studying theology, which she started while she was in Stuttgart and Freiburg. And she's currently working on a PhD Phil in theology in the University of Oxford. And she's been looking at exploring the theological aspects of viruses. So in fact, she's ideally equipped and especially qualified to speak to us today on the subject of viruses as part of God's creation.
Good afternoon. It's my pleasure to share some thoughts about viruses today. We have already heard a very interesting talk yesterday about how we as Christians can respond to pandemics, with some very helpful thoughts on that. Today I'll talk more about the virus point of view and the biology about viruses, so we can have a discussion about how viruses actually fit into our Christian worldview afterwards. The title of my talk is Viruses as Part of God's Creation ? How do we respond to viruses? I think that's the main topic we're facing this year. We all thought about coronavirus a lot, but actually there are many more viruses that in the past we had to respond to, and there will be more viruses in the future. So I'm thinking of Ebola in 2014, SARS in 2003. We had the Spanish flu, the bird flu, we had MERS, and Zika virus in 2015. In an article that I've co-authored together with friends, we quote a Chinese saying, "If you know the enemy and know yourself, you need not fear the result of 100 battles." But is this a Christian response to a pandemic? So let me quote Martin Luther. He had the same issue that we are having today. How do we deal with a pandemic? He was facing the plague. And as back in the days, we also asked the question, well, if that's God's punishment, why should we respond? Is there a need for us to respond? So what Luther was saying is, they say that it is God's punishment. If he, God, wants to protect them, he can do so without medicines or our carefulness. This is not trusting God, but tempting him. God has created medicines and provided us with intelligence to guard and take good care of the body, so we can live in good health. What it means is that it is a very healthy Christian response to try and understand the biological threat, to see where we can help, but also then think about the implications of this from a theological perspective. So what is a virus? What do we actually know about viruses? At a first glance, viruses do a bit look like the Death Star in Star Wars. This is an example of Zika virus. Flaviviruses do look like that. But when we look at more different viruses, we can see there are actually a variety of shapes they come in. So when you think about rabies viruses, that does look a bit like a bullet. Or Ebola does look a bit like a little snake or worm. They also come in different sizes. So here you can see how they relate to a human red blood cell, and you can see how tiny they are, with the exception of the mimiviruses. That is one of the biggest viruses we know and only recently discovered. But generally, viruses are tiny. Look at rhinoviruses, which causes the cold, so hepatitis B virus. Even HIV is tiny. As researchers, we try to classify viruses into families, and we usually organize them based on their genome. So there's a taxonomy for viruses that divides them into, are they having a genome that's based off DNA, or is it made of RNA? And in the case of RNA, we further divide into double-stranded RNA, single-stranded RNA, and so on. So it is quite challenging, and with every new virus, there is the classification, of course, needs to be added. What do they all have in common when they are so different? So viruses are non-living parasites, and that's quite interesting. So they do not have cell membrane or their own metabolism, which means they actually lack the tool to self-reproduce outside a host. They need us, otherwise there's no reproduction. So when you think about a jar of nutrients and you have a bacterium and place that in it, you can see that the bacterium will reproduce because it is a living parasite, whereas the virus won't. The virus will just sit there and wait. So how does a virus then replicate inside our hosts? Let me show you a few key steps and challenges a virus has to face. So a virus needs to enter our cell, which means that first of all, it needs to pass some barriers like our skin or the mucus that surrounds some cells. And then this entering process is actually also not a passive process. One of my friends lately said that the virus knows which doorbell to ring. So there are some receptors the virus can bind, and they then basically take him up into the cell. A key step a virus has to do is evade the host's immune system. So it tries to hide as good as possible. The virus will replicate the genome, that's the main challenge, but also produce components for new viral particles. In the later stage, the virus then builds the particles, or these particles assemble, and then the virus exits the cell again. So these are the stages the virus goes through. How does it do that? I mentioned it's a non-living parasite. So what is it? I've mentioned there is a genome, And this basically carries all the instructions to build more viral particles. One of the main components of this genome are the ones that will build the virus particle, like glycoproteins. These are on the surface, and they are the ones that attach to a cell and basically ring the doorbell. There's also a bit of packaging involved. So the genome also codes for proteins that will build a shell around the genome, for example. And the last bit, that's quite depending on the virus. So you have smaller and bigger viruses, and the amount of these tools differs between them quite massively. But there are some parts the genome codes for that are not necessarily even packaged into the virus particles, but these are important tools to interact with the host, to interact with whatever we have in our cells to produce viral particles and to block immune responses. Whenever a virus enters our body, there is a fight with our immune system. And we call this arrangement of tools we have an arms race, because both the virus and the host are continuously working on making their tools better to win the fight next time. So what kind of tools do we have? And what kind of tools does the virus have? So in case of the virus, there are many, many things a virus does to circumvent what we offer. So first of all, there's fast adaptation. Viruses don't have proofreading functions when they copy their genome, which means they produce a lot of mutations in the copying process. Because the virus is not a living thing, it doesn't really care. And one of these mutated viruses will be successful. And that's the one new virus that will be passed on. Viruses can also reassort. That means, for example, if you're infected by 2 different influenza viruses, because influenza virus has a segmented genome, these segments can be exchanged and build totally new viruses. Viruses also go into latency. We know that from herpes viruses, for example. So they hide in our cells for quite a long time and then suddenly reappear. Some viruses can integrate into our genomes. We know this from retroviruses, like HIV, which means that we're not getting rid of them at any point. They're just part of us now. Viruses are also good at mimicry. Especially bigger viruses like herpes viruses are known to produce proteins or other things that look very similar to what our cellular components look like, and that makes it much harder for us to detect. And as I've mentioned, a virus produces tools that can interfere directly with what we have in our cells, like our immune responses. So what do we do? Why do we not just all die from all the viruses that infect us? Because viruses are apparently quite good at using their tools. So we have 2 main responses, one called innate immunity and the other adaptive immunity. At the first stage of an infection, basically hours after a virus enters the cell, minutes to hours, the innate immunity kicks in. And that's an extremely rapid but non-specific reaction. So what happens is that we have several sensors floating around our cells, and they detect foreign material. They detect that there's something that's not quite right and start a cascade that will lead to the production of signals that can alarm and inform all other cells around them that there's something that's dangerous. These warning signals, for example, are called interferons. There's also a whole range of other cytokines and chemokines produced. But they set an alarm, and this alarm means that all the cells, the one infected and the one not yet infected, will produce a range of proteins that can directly fight viruses. We know of more than 300 so-called interferon-stimulated genes, and that's basically classifying everything that's upregulated at this stage that can potentially inhibit the virus. Most of these have several functions, and some of them will be successful in eliminating the virus. So that's a very unspecific but very quick response. About a week after we got infected, the adaptive immunity is ready to go. So thanks to the alarm system, B cells and T cells get activated, and they arrange the production of antibodies, for example, which are highly specific and can fight a very specific virus. And the important thing is they also build out long-term memory, which means if we see the same virus again, there's already an immune response that's specific to that virus that can be activated. So there's quite a lot going on when we're infected by a virus. But I also need to mention that there is a dark side of our immune system, like in Star Wars. I've mentioned these interferons, signaling molecules that alarm everyone around them. Like interferons, all of the cytokines we produce, all signals need to be closely monitored, and they also need to be balanced. Because of course the risk is, if you produce too many of these pro-inflammatory responses, the chance is that you clear the pathogen quite well and quite fast, but there's also an increasing risk of tissue damage. Whereas on the other hand, if you have an anti-inflammatory response, so you balance the pro-inflammatory response, that means you protect your tissue from damage, but there's also an increasing risk that some of the pathogens will survive. So a lot of the symptoms, for example, that we face when we have a cold, headache, fever, running nose, cough, tiredness, body ache, all of these symptoms are actually not produced by the virus, but are a symptom of our immune response. They are side effects of our own cytokines. And that is important to keep in mind, because that makes many viruses look to us very similar, although the virus is quite different. But the outcome of an infection is determined to quite a large extent by our response. And I just want to mention that we currently can also watch this. So when we're thinking of COVID-19, we see that there's a difference between men and women and how they respond to the virus. We can see that men are much more severely affected by COVID-19 than women are. And that can, to a large extent, this depends on our immune responses. So let me show you 4 examples. So things we know that are different between men and women and that affect COVID-19 are, first of all, virus entry. So the virus needs a receptor, and the main receptor is called ACE2. And this is downregulated by estrogens, which means that women have less ACE2 on the cell surface in comparison to men, which makes it more likely that a higher amount of viruses will infect men. Second stage, virus needs to be detected. And I've said there are tons of sensors in our cells that will recognize foreign material. And we know of one, which is called toll-like receptor 7. And this is X chromosomal linked, which means that women produce much more of the sensor than men. So there's potentially a difference in a very early response to viruses between men and women. I have mentioned that then there's a cytokine production, which recruits cells like macrophages, NK cells, neutrophils, monocytes, and also the amount and the variation in between the chemokines and cytokines we produce is different between men and women. And lastly, our adaptive immune responses are different as well. So we know from flu infection that the amount of B cells and antibody differs between men and women, but also the T cell repertoires and the different T cell subsets. So there are a large amount of differences between immune responses that actually determine the outcome of a virus. And in many cases, it's not so much the virus. So let's look a bit broader. We've looked at what happens when we are infected by a virus, but what is actually happening to the world around us? So we usually think about viruses in terms of one virus infecting us, then we pass it on to the next person. You're thinking very anthropocentric around viruses. But there are more and more attempts to think globally when it comes to viruses, because viruses do affect the whole ecosystem around us as well. And depending on how our ecosystem reacts to the virus, this will also affect us. So coming back to SARS, coronaviruses have their origin in bats, which means there's a large amount of different coronaviruses circulating in bats. Usually that doesn't harm any of us. The problem arises when one of these coronaviruses gets transmitted to an intermediate host and then is able to jump from that intermediate host to humans. So we've seen that for MERS a couple of years ago. In that case, the intermediate host was a camel. We've seen that for SARS, where it probably was a civet cat. And for SARS-CoV-2, we're still not sure, but it might have been a pangolin. So living with these intermediate hosts will affect the amount of viruses we see. And that's not specific to SARS. So look at this. This is what we know about influenza viruses. Influenza viruses do circulate in aquatic birds, in poultry, in pigs. And depending on what happens in these species, there will be new viruses that we are faced with as well. And there are certain reasons why we don't see that happen too often, but only once in a while. And that's of course because the virus needs to adapt to new host machinery, and it also needs to adapt to new immune responses every moment it basically moves from one species to another. So every couple of years we have a virus coming from a zoonotic reservoir like this year. But the amount of viruses that could actually do so is much higher. And so there are massive thresholds that still protect us. Nevertheless, we live with other species on this planet, and depending on the species and how we treat them, in which way we're in contact with them, this will affect the virus repertoire we see. Right, so for everyone going on a beach holiday this year, here's what you can plan for. So there are about 3 × 10⁶ viruses in the deep sea. There are about 1 × 10⁸ viruses per milliliter in coastal waters, which makes about 1 × 10⁻30 viruses in the ocean in total. Viruses are the most abundant biological entities on this planet. There's not a place on Earth where you won't see a virus if you're looking for it. And we're just learning to understand how many there are and what types there are. So there are modern technologies that help us get an overview. So with metagenomic viral discovery, we can actually sequence what's around us. We can take samples from our environment, like the ocean, sequence and compare to what we already know. And the likelihood is that we still discover tons and tons and tons of new viruses that we've never seen. What does that make of a difference for us and for our ecosystem? I've shown that viruses in other species will at some point potentially infect us, and we need to be prepared for that. But also, viruses do change the ecosystem around us, and the world we currently see the way it is might look really different if the virus load was different in only one of these species. Let me explain that. So you have one individual infected. Potentially, this can change the behavior of that one infected animal. This will change potentially birth rates and death rates, or how a population around this one animal behaves. This will affect the whole community around them, predator-prey relationships, competition around food. And very downstream, it will also affect how the whole ecosystem looks like. Let me name some examples. We know, for example, that sea purfish, they do change their thermal preferences when they're infected with a specific virus, which means they change location. We know of invasive fire ants. They change their dietary requirements, and they're going for a much more carbohydrate-rich diet once they're infected with another virus. And there was an example in Tanzania where they had a rinderpest infection of wildebeest and buffaloes. There were less grazing pressure on the woodland around. There were more fires as a consequence of that. And suddenly the whole environment changed into a grassland steady state. The moment they introduced the vaccine for these wildebeest and buffaloes, the whole thing changed back. So there you can see that one infected individual will have implications for a population, for a community, and the whole ecosystem. And that is highly interesting because we don't know which percentage of the world we currently see is actually determined only by viruses. Let me show you another example. This is the cycle of CO2, carbons. And we know that plankton is highly infected by different species of viruses. And these infections by viruses actually increase the lysis of plankton, which makes carbon more available in the upper or surface areas of the ocean, and it doesn't sink into the deep sea. That not only affects the CO2 levels in our atmosphere, it affects the form of available nutrients for everything that's not in the deep sea, and it changes the termination of algal blooms as well. And we can see a similar thing when we're looking at soil. So there are many polymers in soil, like sugar-based polymers. And we know that there are certain viral glucosidase hydrolyses, and they break them down into single sugars. And of course, that changes fermentation and respiration processes downstream, also production of CO2. But in the very end, the viruses that affect bacteria in the soil will affect the nutrients around them. And this will change the whole ecosystem that lives in that area. So we thought about ecosystems, but there's another point I want to make. Viruses are also driver of evolution. So current research estimates that the development of multicellular eukaryotes was only possible because the world started off with pre-cellular replicators. And I'm not going into detail of what a DNA world or RNA world looked like, but these pre-cellular replicators that later gave rise to a virus world also was the thing that made the development of cellular organisms possible in the first place. So we wouldn't be around if it wasn't for viruses. And it also means that we have co-developed together with viruses. And this has more evolutionary consequences than we usually think of in our daily lives. So a lot of our cellular machinery, for example, is shaped by this co-evolution with viruses. So here's a study that found that viruses are actually a dominant driver of protein adaptation in mammals. They estimate that about 30% of all adaptive amino acid changes in parts of the human proteome are due to viruses. What does this mean? I have told you that viruses interact with our host machinery, and of course the host wants to evade that, wants to escape. That means that as long as viruses are around, there's a constant pressure for our host cell machinery to develop in a way that can evade viruses. So the proteins in our cells, as we know them right now, the majority will have encountered a virus at some point, and about 1/3 will have changed accordingly. That's what the study tells us. There's another way viruses change our cell machinery. So here you're looking at the graph that shows you the percentage within the genome that is made of a specific signature of retroviruses when they integrate. So about 8% of our human genome consists of this retrovirus sequences. What does that mean? I've told you that retroviruses like HIV, they integrate into our host genome and they stick with us for the rest of our lives. And this happened throughout evolution like many times. And there are several consequences. First of all, of course, we can express the original virus and infect other species if this virus builds particles. Of course, we can also express some sort of recombinant virus, which means we can infect other species with a virus that contains slight changes because the virus picked up parts of our genome on the way of building viral particles. But in the last 2 points, I'll go into more details in a minute. There's more to this. Every virus genome, of course, has also regulatory sequences, which means that if these regulatory sequences are very close to genes in our genome, they will affect or can affect gene expression. This means these regulatory sequences become our regulatory sequences. And of course, viruses do bring different proteins. And there are very many examples, where we can see how we actually made use of a viral protein for our own cell machinery. Let me show you one example. So this is syncytine, and syncytine got introduced not only into humans, but also in different other species many times during the course of evolution. And what it does, you can see here. So originally, the syncytine is a surface protein of this retrovirus, and it basically facilitates the fusion of the viral particle with the cell. So this syncytine integrated into our genome. And what it now does, it's expressed in our cells and can help facilitate the fusion between two different cells. So how is that important? we know that the syncytium that is needed when our placenta forms can be facilitated by the syncytine, which means there's a viral protein that got introduced into our genome that now carries a major function when it comes to human placental morphogenesis. I have mentioned that we can also use regulatory sequences that might have been part of viruses and that integrated. So we all need to regulate the genes that we express. And there's one rather labor-intense mechanism, where it basically means that for every gene that we want to express, we need to invent a new model of regulatory sequence and then protein-protein interaction to express the gene. But what if, and that's a hypothesis, what if through the introduction of certain retroviral sequences, because these sequences can copy and paste and integrate in a new place, they already facilitated for us this combination of regulatory sequence and a protein that binds, and enabled us to regulate whole networks of genes at the same time by this copy and paste mechanism. So there's an option where viruses might have changed the way that we as a very complex organism function. Last part of my talk, I just want to give you an estimation of how many viruses and virus families can we actually expect around us. So there's one study, and they looked at the Indian flying fox. They collected samples, and then they found there are 58 different viruses in that sample, and they found 55 of them were actually newly identified. So we only knew 3 of them. So if we think of the amount of mammalian species we know, that means there might be 320,000 viruses that we haven't even heard of. If you think of all the vertebrates we know, there might be more than 3 million viruses that we have never heard of. If you think of all the known species, there might be 100 million viruses that we have never heard of. And this is actually an underestimation, because in this Indian flying fox, they only looked at 9 viral families. And of course, this does not include viruses that can affect bacteria, archaea, or other single-cell organisms. To sum this up, virology is a highly interesting field, and there are new discoveries around the corner for the next hundreds of years. And you can see in the cartoon, question, what is that? It's the virome, and it's moving fast. Is this good or is that bad news? And the answer is yes. So let me summarize this. What do we know about viruses as part of the biological world? We know that viruses are non-living, obligatory intracellular parasites. We know that we encounter them on a regular basis. And we also know that our immune responses also determine the outcome of infection. It's not just a virus. We know that all species on Earth are affected by viruses. We know that viruses are involved in all processes and all ecosystems. We know that viruses shape the machinery of host cells, and they contribute genetic novelty. And we don't yet know how many viruses there are. We're only beginning to understand their contributions to our lives. What does this mean for us as Christians? Viruses as part of God's creation? I think we've seen yesterday that we have to acknowledge the individual suffering, and there are many ways in which we Christians can do this. Medically, as counselors, but also just in relationship to other people. But, and that's the point I want to make today, there's also a global contribution of viruses to the world we live in, and that includes our own existence and our own bodily functions. And we need to be aware of them. And this will open up a dialogue about the complexity of what we declare as good and evil, complexity about answers to suffering, how we as human beings are connected with all living and non-living beings, and the complexity of what it means to be human as well. I will end my talk here, and I hope I could show that biological findings about viruses clearly leave us with very many challenging conversations about highly complex theological topics. Thank you so much for listening, and I'm really looking forward to your comments, your questions, and a discussion around this.
Thank you, Miriam, for a very educational talk. Certainly increased my knowledge of viruses by 100%. But it started off very small. Thank you very much indeed. So we actually have a question kind of left over from yesterday about the effective viruses, in particular the coronavirus that we're dealing with at the moment. The question was, is there a reason in virology that can explain why older people seem to be more affected by the virus than younger people?
Yes, that's a very good question, and also not unique to coronaviruses. So we know this from several viruses, including influenza virus, polio, measles, herpes viruses. So there's a whole range of viruses where we see this, that an increasing age is actually a disadvantage when it comes to fighting the virus. And there are several reasons for that we know of. And I think there are a bunch of reasons we don't yet understand, because there's a lot about aging that we don't understand. But so some things we know is that when you're infected by a virus, it's not only the virus that determines the outcome or the amount of viruses you receive, but it's also about the state of your immune system, for example, and your gender, and I've mentioned that. So there are hormonal changes, and also with increasing age, your hormone levels change as well. There are differences in your metabolism when you're aging. We can see this, for example, with an increase in diabetes in older people. And also for coronavirus, we know that diabetes is negatively correlated with the outcome of infection. And we also know from virology that it also depends on in which order during your life you see a virus first. So for flu, we call this antigenic sin, which basically means your immune system starts memorizing the pathogens, and depending on which one it sees first, there's a predisposition also of your immune system. So for example, in 2009, we had the swine flu, and interestingly, the older age group was more protected than the middle ages, and that depended on the flu virus that they had seen before, because there was still a small amount of H1N1 that caused the Spanish flu in 1918 circulating when they were growing up. So they had seen that one before, and that then, although the virus was slightly different, that still partially protected them. So we see this for other viruses as well. And I think for coronavirus, there's still a lot of research coming the next couple of years, because it's one of the most interesting bits for coronavirus, since the virus doesn't change a lot, but our immune responses are quite different depending on who gets the virus. So I think this will be a major topic of research in the future.
The differences between the way male and female people react to the virus, how it's affected by them. Is there any indication that there are different responses from different genetic or different racial types?
So I have not seen a publication on coronaviruses and racial differences. What we know from other viruses is that part of this innate immune response, these very early genes, expression levels change with so-called single nucleotide polymorphisms, and they can be racially inherited in some way. So we know in the Chinese population, for one of these ISGs, there's a SNP, and that depends on that one. You can see how flu is affected. But I think that needs a whole lot more data and more in-depth studies for coronaviruses to see what the impact of that is.
Right. As far as the mechanism is concerned, the question has been asked, when the virus rings the doorbell, as it were, at the cell, as you put it, and there's only one way in that the virus can enter the cell, is there also only one way the replicated viruses can escape from the cell, or how does that happen?
So that depends a bit on the type of virus. So some viruses are lytic viruses. That basically means they kill the host cell, and the cell dies. And while the cell integrates, the viruses can exit. So that's the more brutal way to exit a cell. A lot of viruses do exit via the Golgi apparatus, because what needs to happen is you have all these building blocks of a new virus, which means a genome, and then proteins that encapsulate the whole thing, and glycoproteins that need to be part of the membrane. And then it all needs to find a membrane and needs to be orientated in the right amount and the right order of things. And then it basically is budding out of the cell through membrane. And so usually it starts with the endoplasmatic reticulum, moves its way through the Golgi apparatus, and buds like other vesicles as well. So that can happen as well.
Okay, thank you. So there was another question about the point you made about the placenta. Can you remind us of some of the positive impacts that viruses have? And does that mean that they're kind of symbiont rather than a parasite in their action?
So I think that's a very subjective discussion about are they parasites or not. So it depends a bit on also on which point in time you look. I think the placenta is a very good example of how our body made use of something that the virus left him with. So there's also a contribution of the cellular machinery to make use. The same with these regulatory events. If you have regulatory sequences of viruses, you might use them, and that might help you. Whereas, let's say, an infected individual who has the wrong regulatory sequence in front of the wrong gene might just not survive. And so these are not basically kept in the human population. So there's an evolutionary component to beneficial effects as well. So by the time you're infected, you might not know that that actually benefits globally, whereas now we're benefiting because someone else had suffered through an infection. So I wouldn't go as far to say it's a symbiotic relationship, but it's clear that viruses have more positive effects than we currently know. And I think a big area of research that is just about to start really, or has started the last, let's say, 5 to 10 years, is analyzing what kind of viruses our ecosystems have and how that actually affects all the species that we encounter on a daily basis. Also how the oceans work and things. I think that there's much more to be discovered. So I wouldn't go as far to say it's a symbiotic relationship, but it's clearly something that also helps create the environment that we live in. So we're benefiting here as well.
Okay. A question here about the work of Martha Clokey. Apparently has done work about viruses that could improve our health. Apparently she spent many years looking for phages that attack the superbug, C. difficile. Have you any comment on that work and how
so
it may be possible?
Yeah, I'm not an expert on bacteriophages, but I think there's some promising examples out there, because what bacteriophages do is they infect bacteria, which might make them a good tool to target bacteria specifically. I'm not sure about health concerns in a sense of something that can enter a bacterium. If that goes wrong, it might also be able to enter something else. So I think this is the threshold here, that we need to make sure that that is safe. But I think generally as a tool, you can use viruses to enter a whole lot of stuff, including bacteria or cells. So there's a lot of therapeutic options out there if you make sure they're safe.
Right. Another question where the questioner has worked on CAMV and TMV, and he wonders if you can say how plant viruses fit into your overall understanding.
Sorry, can you repeat that question?
The question is, what's the role of plant viruses in the overall understanding of their role in creation?
Also not an expert in this, and I might need to get back to you about that on Friday. What I know is that it's basically a similar effect as with other species. So if you're infected with a virus, this will change how an organism works, and this will also change how plants work. And then you have two types of plants. You have the wild living plants, and then you have the ones that we use for agriculture. And I think there's a difference there on how you judge if the virus is actually beneficial or not. So in wildlife, I think it's currently still hard to tell, because we don't have enough data on enough plants and enough viruses there, I would assume. With the agriculture, I think that's more urgent, because we need to feed a lot of people on this planet. So I would need to look into research what's going on there, because I'm not an expert there.
Okay, so we're going to move on to something more theological slant, as it were. Miriam, I wonder if you could comment. The question is asking, can you see the hand of God working through viruses?
That is a very good question, and also a very philosophical one, because of course we can't know what God is thinking in that sense. You know, did he create viruses as a tool, and what we see now is basically the result of that, or are they something that he allowed the freedom to develop, and we're living with what happened with that, which I feel is the more likely scenario, that there was a lot of freedom in how this world developed. So it was maybe more of a passive tool than an active one. But I can see a lot of beauty in viruses when you look at pictures and images, just how they optically look. There's symmetry, there's beauty in there. So I think that is something we sometimes underestimate of when we think about viruses, because we're usually thinking about the functions, but there's also beauty in the way they look. And then you think about the functions, and it's quite interesting how they mechanistically work. And I wouldn't say that God is not. I would not say that God is not using them or can't use them. Clearly, they're left for us also as a tool. Since we do more research, we can use them as a therapeutic agent in some way. So I wouldn't say that they are more or less a tool from God or a gift from God than other things we see on this planet. We have other beautiful things, and depends on who you ask. If you ask Bob, who gave the talk yesterday, he might agree that viruses might be a fun way for God to act, but I think he has other suggestions of what he thinks is more exciting, depending on his research topics. So I'm quite excited about viruses. So I can see God in viruses just because they're beautiful and mechanistically intriguing, and there's a lot that developed through viruses. If you ask someone else, they might have different opinion on that. So I think I'll leave you with a very subjective note.
Okay, well, I'm not going to let you off with that completely, Miriam, because there's another question from, if you like, another angle. And she asks, do you think pathogenic viruses could have been produced by Satan interfering in the geological ages with the evolutionary or other processes God was using to form the world for human habitation?
So there's some, yeah.
So was Satan or the spirit of evil actually interfering with God's creation by making bad viruses?
I think there's no such thing as a bad virus, because I think most viruses can have beneficial and not so beneficial effects depending on who's infected and who you ask if they're benefited or not. So for example, the coronavirus pandemic did have some benefits for other species, not for us, because we are the ones sick and dying, and we are the ones quite limited in our lifestyles right now. But I think other species are benefiting currently. So this basically raises the theological question: if we attribute this disaster for us to Satan, then we would also need to claim that Satan would actually help other species at the same time. And then you're coming into theological difficulties with what do we think about the rest of the world we look at? Earthquakes created an environment that we are benefiting from. If we claim that all these evolutionary pressures that led to the formation of humans, if we attribute them to Satan, then there's little left for God to be responsible for. And so I think there's this weird tension of beauty and benefits that goes along with suffering at the same time. And if you state too strongly something's only caused by Satan, then then there's this issue of if we can't really divide them by God and Satan. I'm not sure if that helps.
In a sense, it's our response to things which in nature may be neutral or would be plus or minus. We have a saying in this country that when you go walking in the mountains, there's no such thing as bad weather, just the wrong clothing. Yeah,
and I think that's an attitude you can have. With the virus, of course, you can't choose by the clothing, because I mean the way your immune system works, your age, your sex, you can't choose all of that. So I agree that there's a bit more difficulty here to explain and a bit more tension. Yes. But it's also not that easy to attribute all evil things, or what we call evil, to Satan, because that takes away a whole lot of creation that we could also think God wanted us to have. So.
Yeah. There's a related question here which asks, could the idea of nature being fallen that we touched on yesterday, but the idea is held by some people that nature itself is fallen, either as a result of human or angelic sin. Could that help to explain why there are negative relationships between viruses and other biological entities?
So yeah, there's one attempt to think that viruses work slightly different and only beneficial before some sort of fall, and then only after the fall started to have these negative effects. Honestly, I'm not sure if we can, from all what we know about biology, if there's a way to claim this biologically, because if you have an agent that causes something, and for most of other agents we know of, like drugs we use, knives we use, technology we develop, there's always the risk of side effects. So I'm not sure if you could have had a virus that is solely beneficial and not harmful for any species. So there's that one response. And then there's the more theological response. What are we sure, or how do I phrase that? The more theological approach is, how do we read the book of Genesis? And I think there's a lot of options of how we interpret that as well. And it depends a bit on if you think of the Bible more as revelation to humans to show how the relationship with their creator looked like and what God intended it to look like, and if that is the main aim of the Bible. Because I don't think the Bible can explain natural sciences. That was never the intention. So I think reading too much into the Genesis book in terms of how the natural work mechanistically worked is taking it too far. That's not how it was written, and that wasn't the intention. And I think if you, for example, if you receive a letter by your doctor about your eyesight, that will be very technical, and someone will think about, that's how far you can see, that's how it works mechanistically. But then you can receive a love letter from your boyfriend, and he just tells you how beautiful your eyes are, and both accounts are true. They just look from a really different angle and have very different intentions. So I think knowing why something was written and what the knowledge back in the days was, I think that is important, because God would not have revealed details about bacteriophages back in the days, because what would people have done with that knowledge? Nothing really. So I think if the Bible would have been written today, it also would look slightly different in what God would allow us to know or understand. So.
Okay. Yeah, just another slightly technical question. I wonder if you could clarify the origins of viruses in evolutionary history.
So that's a really interesting topic, and also I think that's still under a lot of debate. So I think classically the attempts were very different. So some people thought that viruses developed before everything else, like as a complete virus. So you had an RNA and then later a DNA world, which was basically just nucleotides, and they started to have some self-replicating features and then accumulated over time, like the capsid, the packaging, the glycoproteins around them. And then after that, that's how eukaryotic cells, for example, developed. And there are other approaches, and they would rather suggest that cells somehow developed, and then out of the cells, something basically budded off the cells that was then able to infect another cell. So these are very contrary opinions. And I have found quite helpful a third approach that tries to combine both of them. And I'm not sure what evolutionary virologists think about that. I need to find someone to actually talk about that. What I find helpful, because I think it explains a lot, is that we have these self-replicating genomes, which I think helped to establish eukaryotic cells in the first place. But because of this interplay back and forth between something that's self-replicating and the cell that it infects, so both the cell and the virus could pick up stuff on the way. And so in this approach, it would be that there was a very basic concept of a virus in the first place, and then they got more complicated on the way. And I think that does make a lot of sense in the way that I see the immune system, and in the way the immune system changes as response to viruses, and as I see viruses respond to our immune system. So I feel that's a very natural understanding of how this whole field developed.
Okay, thank you. I think we'll make this the last question. Miriam, thank you very much so far. I wonder if you would be prepared to tell us a little bit about your own response to the COVID pandemic, both personally and spiritually.
So I think there's always two sides of this. There's me as a virologist, and of course for me as a virologist, this is highly interesting. in a sense of there's a virus that reacts differently with our immune system, and there's an immune system that's very naive and tries to figure out how to deal with this virus. So I think there's very interesting research currently done, and even more interesting research in the future that we can look forward to. So I think that's quite intriguing as is. Plus, I had lots of very interesting conversations with friends and family on a level where we've never been in our relationships, because of course they know what I do for a living, but in so much detail they had never asked, and I wasn't. Yeah, I didn't throw wisdom on them, but this was a very interesting time to explain what I do for a living, and to explain what I like about viruses and what's interesting, and also what's difficult, and help them understand how research works, because I think that's also, for most people who are not used to work academically, just understanding the process of experiment and then result and then discussion and then another experiment. And because that was so public, I think that was really interesting on that level. I think personally, of course, I think I went through similar stages as everyone else. I was frustrated of being locked in. I was suddenly limited in a whole lot of things that I love doing. I was limited or had to express my relationships in different ways. On the other hand, I also gained a lot of time. I could focus and reflect on different levels, especially theologically, about viruses, because I was watching a pandemic. I was getting more responses, and I had to think broader and wider. So that was also theologically stimulating for me. I've seen people suffer. I've seen people lose friends and family. So I think there's also a big component of grief on relational levels. There's a big component on grief in a sense that I've lost things that would have happened that are not coming back. So I think these things I've experienced in a very similar way as everyone else. And I think spiritually, I discussed this with my church last week. I was also going through a journey because I think there are two things that are interesting. First of all, we suddenly have more time with Jesus by ourselves, because we are now sitting in empty rooms and don't have a whole church community that we can physically meet with, or at least didn't have for a long time. And so I think there was a certain strength and beauty in that, having more time in a personal relationship with Jesus, more time to read my Bible and to pray by myself, which usually in my very busy day, of course, is cut rather short. So that was a beauty in that. But at the same time, I started to feel the loss of the community around me. And I think being a church means being with other people, journeying together and being alongside. And I feel, although we have a lot of technology, I'm still grieving that loss. So I think, yeah, that's what happened to me spiritually.
Okay, thank you, Miriam. I'd like to thank, on behalf of everyone who's been listening, thank you for your talk, and we've learned so much. Thank you too for the way you've answered our questions, and perhaps to everyone else, if you have more questions, do feel free to send them in on the same routes through the comment box or through your social media channels, and we'll try to get to them in our panel discussion on Friday. But before we say goodbye until tomorrow, I just want to remind you again of the opportunity that you have to help us finance the conference and the ongoing work of Christians in Science. And you can phone and pay by text using Give to CIS. The details on how to give will be on the screen here and afterwards on screen and on our website. There will be a standard charge for the text that will depend on the network that you're using. Could I also remind you and encourage you to send in your feedback form. It takes only a few minutes to fill in, and it would be really helpful to us, and we'd love to hear from you. So find the feedback form on our website or in the video feed. And remember, you can also follow us on social media, on Facebook, Instagram, and Twitter, and find out more about us by visiting our website. That's www.cis.org.uk. So thank you again for being with us today, and we look forward to you joining us tomorrow at the same time when Dr. Simon Kolstow will be speaking about the ethics of evidence in a pandemic. Goodbye.